Hi students, can you hear me? Please let me know if you can hear me so we can start the session. Can you hear me? Okay, so hi everyone, I am Dr. Devyani Puri and today's session is going to be about the bacterial growth curve. So this is super important when it comes to your undergraduate classes, but also very important for the postgraduate examination because you do get a lot of questions from this particular uh, topic. So bacterial growth curve, let's start. So as we all know, that the bacteria has a particular curve of growth, like all of us humans also do. So what do we do first? Whenever we have a solid media, so suppose we have taken some bacteria and we inoculate it into a solid media, right? So we have inoculated this bacteria onto a solid media, and what are we doing with it? We are letting it grow. So once we have these colonies, can you see these colonies? So once it has grown on the solid media, then we take up these colonies and we put it into some liquid media because the bacteria has maximum amount of growth in a liquid media. And if you want to study the growth curve of the bacteria, it's best to inoculate it into a liquid media because this liquid media contains a lot of nutrients that is required for the bacterial growth, and hence this is liquid media is required. So we have inoculated this colony into this liquid media. Can you see this? And we have given all the nutrients that are required for the bacteria to grow. Next, what do we do? We incubate this tube or the liquid media at 37 degrees Celsius because as we know that bacteria needs this optimum amount of temperature, which is 37 degrees Celsius for its growth, right? And then the bacteria grows and follows a path. So what is that path? So this is what the bacterial growth curves looks like. So there are mainly four phases in the bacterial growth curve. So one is your lag phase, right? Then there is a log phase, there is a stationary phase, and then there is a decline phase. So it all this depends upon the number of bacterial cells in that medium, and how much time is it going to take, how much time they are taking to go through all of these phases. So there is a lag phase, log phase, stationary phase, and then a dead phase, and also we call it as a decline phase, right? So four phases. I'm going to talk about them individually later. So next I want to talk about what is a bacterial count. So what is actually a bacterial count? So we will divide this into two parts. So the first thing is going to be a viable count. So what is a viable count? Viable count is nothing but all living bacterias. So all living bacterias in a medium is known as a viable count. Viable means living, right? So all the living bacteria in a medium is known as a viable count. And this viable count is actually dependent on two major things. One thing that it is dependent on is, so it is dependent on if there is division taking place. So is division taking place? So if the division is going to take place, so the viable count is going to increase because obviously the number of progeny is increasing and those progenies are living. So it's going to add up to the viable pool, right? So if the division of the bacteria is taking place, the number of progenies, living progenies are increasing. So the viable count is going to increase, right? And the next thing that it is dependent on is the death. So what does this mean? This means that if death is increasing, that means if death is increasing, if the number of people or the number of bacteria that are there are dying, so what will happen to the total pool of the viable count? So number of living people are going to decrease, right? So if death increases, the viable count decreases. So these two things you need to remember because these are some things that are asked in your examinations as well, right? So what are they dependent on? One, if the division is taking place, if division is taking place, the viable count increases, and if the death increases, the viable count decreases, right? So that is the first thing that I want you to know about the bacterial count. So now let's come to the second thing. What is the second thing? The total count. So what is the total count? So total count is nothing but the total count of the viable plus the dead bacteria. So in this, it's going to add up to both viable and dead bacteria, right? So this is also important because as we go further, you'll know that in which stages or the which phases of the bacterial growth curve this total count is increasing or decreasing, and will all depend upon if there are viable organisms or if there are more dead bacteria in that particular medium, right? So next, let's talk about the four phases. So as we are seeing here, what are the four phases that we talked about? So there is lag phase, there is log phase, there is a stationary phase, and then there is a dead or the decline phase, right? So first we are going to talk about the lag phase. So here I want to actually tell you a story so that you can remember it like this. So lag phase is nothing but let's try, like bacteria are also living, right? So they are very much similar to humans and all phases that we go through. So in the lag phase, what you're going to remember a pregnant woman. So here the bacteria is actually acting like a pregnant woman, right? So what is going to happen here? So what happens in the lag phase? So what does a pregnant woman go through? As soon as she knows she's pregnant, she's trying to adjust to the environment, right? She's trying to know what is all around, how to go about this pregnancy. So in the lag phase, the bacteria also adjusts to the environment. Right, so the bacteria is also adjusting to the environment in this phase, right? It's trying to get used to that, okay, the bacteria is also pregnant and all, so it's trying to adjust to the environment. What is happening next? So, in a pregnant woman, what happens? She needs a lot of nutrition, right? So, here also, bacteria takes up maximum nutrition. The bacteria is taking up maximum nutrition. It has to eat a lot to, produce babies. So here the bacteria is also eating up a lot of the nutrition that is being provided in the medium, right? So that is the second thing that happens in the lag phase. First, adjusting to the environment, adjusting to the news of the pregnancy. And second thing is that bacteria is taking up maximum nutrition because it needs to support this pregnancy. Next, what happens is, so as the woman is growing in the pregnancy, so what is happening? The size of the woman also increases, right? So here also the size of the bacterium, so the size of the bacterium also increases. In this phase, the size of the bacterium is also going to increase. And at the end of this phase, that means at the end of the lag phase here, right here, the size of the bacterium is maximum at the end of the lag phase, right? The size of the bacterium is maximum at the end of the lag phase, right? So this is what the lag phase is. You need to know. Remember, a pregnant woman, bacteria is adjusting to the environment. It's taking up maximum nutrition. The size of the bacterium is increasing. And when is it reaching the maximum size? At the end of the lag phase. So this is what lag phase is, right? So next we are going to talk about the log phase. So now what is going to happen in the log phase? Again, what are we going to remember? Now this pregnant bacterium is going to deliver, right? Now it's reached the phase of delivery. So delivery of the bacterium is going to happen. So what is going to happen here, right? What is going to happen here? First, the progeny, right? The number of babies are going to increase. So the progeny is going to increase. So exponential growth is going to happen. So exponential growth is going to happen. So as we see here, what is happening? It is exponentially growing back to back, back to back. So much growth is happening, right? So exponential growth is happening because here now the bacterium is delivering and producing a lot of babies. So one, the progeny is increasing and exponential growth is happening. Next, what is going to happen to the mother? Now, what is going to happen to the mother bacterium? Once that bacterium has delivered, what is going to happen? The size of the mother. She's going to decrease, right? Because now she's given birth. So now she's going to decrease. So now here, the size of the bacterium is going to decrease. Correct? So size of the bacterium is going to decrease. Again, what is going to happen? What is going to happen to the viable count? What did we talk about to the viable count? So if the division is increasing, so the viable count is going to increase, right? So is the division increasing in the log phase? It is increasing, right? So now what is going to happen to the viable count? So the viable count here is going to increase because the number of progenies are going to increase, right? So viable count is going to increase. And I want to let you know one more thing, that all the viable bacteria, whenever we are trying to stain, like suppose we are trying to do some level of gram staining or any kind of staining, what is happening to the viable bacteria? So the viable bacteria are takes up maximum amount of staining. The good staining that we talk about is taken up by the viable bacteria. So here, if we want to do any kind of a staining process, right? So here, staining is best done in this phase. So staining it is best in this phase, right? Another thing, whenever we are trying to do a lot of biochemical reactions or we are trying to see which organism produces what kind of biochemical reactions, so exactly here in this phase, because there is so much of the viable count, so biochemical reactions are also best in this phase. Right? So biochemical reactions are also best in this phase, right? So what is going to happen here? We are going to say that the bacterium is metabolically active here. So the bacterium is maximally metabolically active also in this phase. So what again, let's talk about the log phase. Remember, the delivery of the bacterium is happening here. The mother bacterium is going to divide. So the number of progenies are increasing, and that is why there's an exponential growth, right? The size of the bacterium is going to decrease. That means the mother bacterium has now delivered, and the size is going to decrease of the mother. The total viable count is going to increase because the number of progenies are increasing, and as we discussed, it is directly proportional to the division. So division increasing, so viable count is increasing. And here what is happening? It is metabolically active. So what is going to happen here? The staining is going to be best here. The staining is going to be best here, and the biochemical reaction. So whenever we want to do any kind of staining or biochemical reactions, we do it in the log phase of the bacterium, right? Gauri, you're asking any favorable conditions. So yes, giving bacteria all the nutrition that is required, like we give it in a nutrient medium where there's peptone, where there's a liquid media, where there is an agar also. Sometimes if we are doing it on a solid media, we are giving them all glucose and we are giving them a good temperature like 37 degrees Celsius. So these are all the favorable conditions. Favorable conditions meaning ki, uh, like for a human also, if you're getting good nutrition, you're getting good air, everything is a favorable condition, right? So that is what I mean by favorable condition here. So that is what I'm providing it to the bacterium in this media, in this particular media, because I want to see the growth of the bacteria also, right? So coming back to the log phase. So I've already discussed the log phase. Then what is the third phase? Now let's talk about the stationary phase. It's a very, very important phase, right? So now what is going to happen here is, let's think this phase is something when the bacterium has reached maybe 50 to 60 years of age, right? So now the delivery has happened, the babies have been born, now the bacterium has reached at the age of 50 to 60 years, right? So here what is going to happen? So here, what are people multiplying or dividing when they're 50 or 60 years having babies? No, right? So there is no division here. So division is not taking place here. Okay, division has stopped. Nobody is having any babies at 50, 60 years. So there is no division here, right? what is the next thing that is happen? What is going to happen to the viable count? So division is not happening, right? But bacteria are still living. So viable count here is going to be flat. It's not increasing, it's not decreasing, it's just flat. So people are living, but no new progenies. Division is not taking place. So viable count here is going to be flat. ठीक है? What is happening to the death count? So, are people dying when they're 50, 60 years? Yes, there are few people who are dying, right? So, why death count is going to increase here. So, there are few people who are going to die, right? So, death count is going to increase here. So, because of that, what is going to happen? What is going to happen to your total count? So total count is what? Viable plus death. Is viable count increasing? No, it's flat. Is the death count increasing? Yes. So what is going to happen to the total count? It is going to increase. Right? So total count is going to increase, but the viable count is going to be flat and the death count is going to increase a little because yes, there are few people who can die at 50 or 60 years of age. So okay, division is not taking place. So as we also grow old, what happens is कि you do realize that so many people get cancers and diabetes and all these things at an older age, right? Why? Because as we are growing, because of the environmental changes or the food or everything, what is happening is we accumulate a lot of toxic products, maybe in the form of eating some kind of foods or having a very bad environment, maybe having bad air. So what is happening in this phase also? Even for a bacteria, as it's growing older, it starts accumulating a lot of toxic products. So accumulation of toxic products increases at the age of 50 to 60 years, and that is the phase of a stationary phase. So stationary phase is actually a plateau kind of a thing. It's like a flat line, right? So this is what stationary phase looks like. So there are a lot of accumulation of the toxic products at this age. Then let's talk about staining. What is going to happen if I want to stain a bacteria at this phase? What will happen is, as I said, staining is also best taken up by the viable count. So what is going to happen? Viable count, viable bacteria are going to take up the stains. But there are also dead bacteria now here in this medium. Now there is dead bacteria also. So dead bacteria are not going to take up the stain properly. So what will happen? I will have a variable staining. So my staining is going to be variable. Probably some good bacteria or the viable ones will take up the stain, but the dead one will not take up the stain properly. So staining here becomes very variable, right? So as again I said, in the stationary phase, what is happening? So we are accumulating a lot of toxic products. This becomes a very unfavorable, it becomes a very unfavorable environment for the bacteria. And what happens when a bacterium goes through an unfavorable environment? It produces spores, right? It produces spores. So spore formation takes place in the stationary phase. Please remember this. Spore formation, why? Because 50 to 60 years में बहुत unfavorable environment मिल रहा है because there is so much toxic products and granules that I'm accumulating. So what is going to happen is spore formation is going to take place at this particular stage, right? And toxic granules I've already talked about. What is going to happen to the nutrition? So as we know, old people at 50, 60 years age, they don't feel like eating, they don't feel like doing anything. So nutrition is also decreasing in the environment. So nutrition is decreasing. And that is why, because this nutrition, now everybody's eaten so much and now reached at 50 to 60 years of age. So what is going to happen? So the nutrition is decreasing. That is why also it is an unfavorable environment for the bacteria. And so it wants to save itself, right? So it starts producing spores. Got it? Another thing that is happening in this stationary phase is that at this particular phase, the bacterium also starts producing some things. So what does the bacterium produce? So it can produce a lot of exotoxins, right? It can produce a lot of antibiotics and it can produce a lot of uh, bacteriocins. So bacteriocins is something that why are these things produced, by the way? Because the bacteria is now trying to fight with other bacteria. So it wants to, so it's like survival of the fittest because nutrition has decreased, spore formation is taking place. Now bacteria wants to produce some things to kill. So it wants to fight with others. So it's going to produce a lot of exotoxins, antibiotics, even bacteriocins. Bacteriocins is something which is produced by one bacteria to kill another bacteria. Right, so bacteriocins, antibiotic, exotoxins, all these things are produced by the bacteria because it's trying to now fight with the others because again the nutrition is deficient. So because of the deficient nutrition, one, spores are forming. The other thing is bacteria starts producing a lot of exotoxins, antibiotics, and bacteriocins. Right, so this is the stationary phase that, so what is happening? 50 to 60-year-old bacterium, there is no more babies happening, no more division is taking place. The viable count is flat because again there is no division, but few people are dying, so the death count is going to increase. Right, so if the death count is increasing, so what is going to happen to the total count? It is going to increase, right? So again, as we are growing older, we are accumulating a lot of toxic products, and because of that, it is an unfavorable environment to the bacteria, so it starts producing spores. And what is going to happen to the nutrition? The nutrition, because then now there are so many people in this age group, so now the nutrition is decreased in the environment, and because of that, the bacteria now wants to protect itself. So it starts producing a lot of exotoxins, antibiotics, and even bacteriocins because it's now trying to fight with others for nutrition because nutrition has become deficient here. Right? Understood up till now? So this is what are stationary phases. So now let's talk about the last phase, which is now the decline phase or the death phase. So here, as the name is suggesting, what is going to happen? Here, the bacteria are dying. So your viable count. So as I said, death increases, viable count decreases. So viable count is decreasing and your death count. So what is happening to the death count? It is increasing. So what is going to happen to the total count? So total count को क्या हो रहा है? अगर viable increase हो रहा है और viable decrease हो रहा है और death count increase हो रहा है तो total count को क्या होगा? It will become flat because they are going to cross out each other. So the total count is going to become flat here. So at this phase, it is the decline phase or the death phase, right? So what is happening? Viable count is decreasing, death count is increasing, total count becomes flat. Again, what is going to happen? इस point पे the nutrition has finished. So nutrition has finished. उधर शायद फिर भी बचा था when it came to the stationary phase. Some level of nutrition was decreasing, but here the nutrition has completely finished, right? So again, toxic products are going to increase, toxic accumulation are going to increase. And at this phase, some bacteria, what do? They start producing certain involutional forms. So what are those involutional forms? Either they will lose their cell wall, right? Either they will lose the cell wall or they will start changing its shape or they will start changing its size, still trying to survive. Even at the decline phase, some bacteria are still trying to survive. That is why involutional forms are seen in this phase. So it is very, very important कि involutional forms कौन से उस पे दिखते हैं? It's in the decline or the death phase. ठीक है? So again, once more revising from the lag phase. So lag phase, pregnant woman has just gotten to know that pregnant, so she's trying to adjust to the environment. Just a second. She's trying to adjust to the environment. So adjusts to the environment, takes up the maximum nutrition at this phase, and obviously the size of the bacterium is increasing because she's pregnant. And at the end of the phase, it's going to be at the maximum size. So that is the lag phase. Delivery of the bacterium is going to take place in the log phase. So now it's going to divide. So now what is going to happen? The progeny are going to increase exponentially. Size of the bacterium is going to decrease. So what is going to happen? If division is taking place, viable count is going to increase. So viable count is increasing and it is directly proportional to the division. What is going to happen here? Maximum metabolically active. So best staining. So staining and biochemical reactions are seen best in the log phase. Next, coming to the stationary phase, what is happening? 50 to 60-year-old person. So definitely not having any more children, not dividing, not having any more children, right? Viable count को क्या हो रहा है? Viable count is flat because no new children are happening, but people are still living. So flat viable count. Death count is increased a little. So what is happening to the total count? Is going to increase, right? And again, as I said, accumulating of the toxic products. So staining is going to be variable because some are dying, some are living. So variable staining. Unfavorable environment, so hence forming spores. And then bacterium produces a lot of different exotoxins, antibiotics, bacteriosis because it has to fight with others for nutrition at this phase. Finally, coming to the death phase or the decline phase, viable count is totally decreasing, death count is increasing, so total count becomes flat, right? And here the nutrition has finished. So again, involutional forms still trying to protect itself. The bacterium is still trying to protect itself. So there are certain involutional forms where they either lose the cell wall, they change the shape, or they change the size. Right. So now we are going to make a table. So I want everybody to make a table so that everything is there. Sorry, everything is there at one place only. So here we are going to write about all the four phases. So we are going to write about the 1 second. So we are going to write about the lag phase, then we have the log phase, we have the stationary phase, and then we have the decline phase, right? So what is the first thing that we are going to write? We are going to write about the division. So is division taking place in the lag phase? No, अभी तो division नहीं हो रहा है lag phase में. Lag phase में सिर्फ nutrients accumulate कर रहे हैं, preparing itself for the delivery, right? Is division happening in the log phase? Yes, it is happening in the log phase. Is division happening in the stationary phase? 50 to 60 years में is anybody having children? No, they're not having children. So definitely not happening in the stationary phase. And decline phase में तो beyond 60 years of age तो okay, decline phase is more than 60 years of age. Let's put it like this. So, decline phase mein what is happening? Decline phase mein is anybody having babies? No, they're not having babies. So, division is just seen in the log phase. So, that is the first thing. So, next let's talk about the viable count. So, is viable count meaning are they is there uh, any new, sorry, is there any new progeny seen in the lag phase? Viable count, what is happening? The ones that were already living are the ones that are living. No new viable count is being added. So here the viable count is going to be flat. The same ones that were already living are the ones that are living, right? So what is happening in the log phase? Is division happening in the log phase? Yes. So is viable count going to increase? Yes. As I said, division increases, viable count increases, right? So so viable count here is going to increase, right? Next, what is happening in the stationary phase? In the stationary phase, is the viable count increasing? Is it increasing? No, it's not increasing. It is flat, right? The viable count is flat. People who are living, no new people are having any kind of babies in stationary phase, so it's flat. And decline phase, what is happening? So the death is increasing. So what is going to happen to the viable count? It is going to decrease. So number of living people, number of living bacteria is going to decrease in the decline phase. So that is your viable phase. Next, death. Let's talk about the death. Uh, is death happening in the lag phase? No, it's not happening in the lag phase. Is death happening in the log phase? No, it's not happening in the log phase. Is death happening in the stationary phase? Yes, some level of death is happening in the stationary phase. Right? Is death happening? Yes. And is death happening in the decline phase? A lot of death is happening in the decline phase, right? So that is what a death, the phases are there in the death ones. So next we are going to talk about the total count. So total count was what? It is viable plus the death count. So let's see from here. So if your viable count is flat and your death is also flat, not happening. So what is going to happen to the total count? It is going to remain flat or same, right? So it is going to remain flat. Again, what is happening in the log phase? Is viable count increasing? Yes. Is death increasing? No. So, but total count is viable plus death. So if viable is increasing, the total count is going to increase. So what is going to happen in the log phase? The total count is going to increase. Again, what is happening in the stationary phase? The viable count is flat and the death one, sorry, the total death count is not so much, right? The death count is also not so much and your viable count is flat. So what is going to happen in your total count? It is going to increase because now here the death is going to increase, right? And what is going to happen in the decline phase? The viable is decreasing, but your death is increasing. So what is it going to do? It's going to cut each other, and here also your total count is going to become flat, right? Understood up till now? Understood up till now? Great. So these were the fours. Now, one thing that I want to talk to you about here is the special features. So what are the special features? We have already spoken. So in your lag phase, what was happening? So there was increase in size, increase in progeny, increase in the viable count, right? Increase in size, and there is increase in nutrition because you need a lot of nutrition to have babies. So the lag count is going to increase. Sorry, the nutrition is going to increase in your lag phase, right? So these are the special features that are happening in your lag phase. What is going to happen in your log phase? Now the baby has happened. Now what is going to happen to the size? It's going to decrease in size, right? It's going to decrease in size. It is metabolically active. So what is happening here? Best staining. Staining is best here, or biochemicals are best here, right? So staining is best here, biochemical is best here, metabolically active, size is going to decrease of the mother. Now coming to the stationary phase. So a lot of things are happening in the stationary phase. One thing that is happening is what is going to happen? What is going to happen in the stationary phase? So here also the viable count is same. So viable count is same. Death is increasing a little. Nutrition is decreasing, right? So unfavorable conditions. So because of that, what is happening? So unfavorable conditions, so spores are formed here. Again, very, very important. Spores are formed in which phase? Stationary phase. So spore formation is taking place. And then what is happening? The bacteria is trying to protect itself, try to fight for nutrition for with other bacterium. So lot of exotoxins and antibiotics and your bacteriocins. So these are going to be produced in your stationary phase, right? And finally, what is happening in the decline phase? Nutrition has stopped completely. Nutrition has stopped completely, but the bacteria is still trying to protect itself, trying to fight. So involutional forms, right? Involutional forms are increasing and a lot of increase in the toxic granules because it's accumulating a lot of toxic granule. So this one table, if you just remember this particular table, all your other things will be answered. This one table is very, very important because this answers all your questions when it comes to the bacterial growth curve. So I want you to make, this table and stick it somewhere so you can see it every time you look at it and then you can remember small small things from it, right? So that kind of sums up today's topic, but I want you to answer certain questions for me. So these are certain questions that were there in your examination of the previous years. So sporulation occurs in which phase? So in what phase is sporulation occurring? Is it happening in the lag phase? Is it happening in the log phase? Is it happening in the stationary or is it happening in the decline phase? What did we talk about? Lag phase में sporulation हो रहा है. Sporulation किस time पे होगा? Whenever there is an unfavorable environment, whenever the nutrition is decreasing, right? So in what phase is the sporulation going to happen? It is going to happen in the stationary phase. Just like we spoke about, sporulation is happening in the stationary phase because in the stationary phase only it's giving a very uh, unfavorable condition and the nutrition is also decreasing. So now in the stationary phase the bacteria will try to protect itself and form a spore, right? And whenever this bacterium is now going to get a favorable condition, like if I take up this spore and put it into a medium that is having good amount of nutrition, so this spore will germinate into a bacterium and then further division can take place. Right? So, yes, Sunny, very good. The answer is C. It's the stationary phase. Right? So, next, this is the question maximum cell size in which phase? So, maximum cell size किस phase में आ रहा है? Lag phase में आ रहा है maximum cell size, log में, stationary में या decline phase में? So, what did we remember that pregnant woman? Pregnant woman lag phase में सबसे ज्यादा खाना खा रही थी, right? She was eating, she needed a lot of nutrition in the lag phase because she had to produce babies in the log phase. So maximum cell size, pregnant woman, which phase? Lag phase. So the answer here is the lag phase. Because lag phase में she's eating maximum nutrition because she wants to give birth in the log phase, right? So maximum cell size is reached in the lag phase, right? Everybody understood? Is everybody on the same lines as me? Great. So next, what are we going to talk about? This third question regarding the lag phase. What is true? So what is true regarding the lag phase? So is time taken? Just a second. Is time taken? Lag phase is the time taken to adapt to the new environment, or is it growth occurs exponentially? Plateau in the lag phase is due to death, and second phase in the bacterial curve. So what is the answer here? So what is the answer here? The answer here is going to be time taken to adapt to new environment. Why? Because as I said, if you remember, going back to where I wrote it, it's trying to adjust to the environment, right? The bacterium is trying to adjust to the environment in the lag phase because it has just found out that she's pregnant and now she's trying to adjust to this new life, that she's going to live. So time taken to adapt to the new environment is right. Is growth happening exponentially here in the lag phase? No, it's not. Sorry, it's not happening in the lag phase. It's happening in the log phase. Babies are being produced in the log phase. Growth is happening in the log phase, right? Next is plateau in lag is due to death. Plateau in the lag is not due to death. That plateau that there is, let me go back to the growth curve. Maybe they're talking about this plateau. So this plateau is not because of the uh, death. It's because it is getting adjusted to the environment. Because it is getting adjusted to the environment and now taking nutrition. So that is why there is a plateau in the lag phase. It is not because of the death, right? And it is the second phase in the bacterial curve. No, it's not the second phase. It is the first phase. So how does it go about? So there is a lag phase, log phase, a stationary phase, and decline phase. So lag phase is the first phase, log phase is the second phase, then stationary phase is the third phase, and finally, what is the fourth phase? Fourth phase is the decline phase or the death phase. Yes, Pallavi, very good. The answer is A for question 3. So, so these are the four phases. They can even ask you questions like which is the third phase in the bacterial growth curve, right? So which will be the third phase? It is going to be the stationary phase, right? Or what is the first phase in the bacterial growth curve? It is the lag phase, right? So I think that sums up my topic for today. It was a very, very short topic, but just please remember this table. Just write it down somewhere. I want you to remember and memorize this table because if you're able to do that, then you will definitely never, never have any wrong questions, from no wrong answers from this particular topic. So if somebody has any doubt, you can please write it in the comment section. The scavengers, I hope it's helpful. My daily micro mysteries are helpful to you. So thank you for that. Any question regarding this particular topic, I'm happy to take it. Else, I am done from my side. So I think we are done. So